Hello everyone, and thank you for attending today's webinar, Metabolomics in Drug Development and Precision Medicine. I'm Martha Powell, a digital editor here at Future Science Group, and I'll be your host for today's event. Before I introduce you to our speaker, I'd like to quickly cover a few housekeeping items. Firstly, all the widgets are resizable and movable, so feel free to move them around to get the most out of your desktop space. You can expand your slide area or maximize it to full screen by clicking on the arrows in the top right-hand corner. A copy of today's slide deck and our feedback survey are located in the resource list, and we encourage you to download any resources or links you might find useful. Please let us know your thoughts on today's webinar, tweeting us at Bioanalysis Zone using the hashtag BZWebinars, or simply share today's webinar with your friends and colleagues using the share widget on your screen. Some networks cause slides to advance a bit more slowly than others, so logging off your VPN is recommended. If your slides are behind, pushing F5 on your keyboard will refresh the page. If you have any other technical problems, you can find additional answers to some common technical issues located in the Help widget at the bottom of your screen. Finally, an on-demand version of today's webinar will be available in approximately an hour after the webinar and can be accessed using the same audience link that was sent to you earlier. So, we're delighted to have Tim Boelke, Managing Director at Metanomics Health GmbH and Chief Medical Officer of Biocrates Group, presenting for us today. Tim graduated as a medical doctor from the Free University of Berlin in 1987 and is a certified pharmaceutical physician at the University of Cardiff. He joined the pharmaceutical industry in 1988, occupying various senior research and development and business roles, before becoming Managing Director of Metanomics Health GmbH in 2009 and recently gaining additional responsibility as Chief Medical Officer of the Biocrates Group. We invite the audience to submit any questions they may have for Tim throughout the webinar using the Q&A widget, which is just to the right-hand side of your screen. We'll pose your questions to Tim in a live Q&A session after his presentation, and we'll follow up any outstanding questions offline. So thank you again for joining us today, and I'll now hand over to Tim to start today's presentation.
Well, thank you, Martha. Good evening in Asia, good afternoon in Europe, and good morning in the Americas. Thank you for taking the time today to participate in our seminar or webinar, Metabolomics in Drug Development and Precision Medicine. As Martha introduced myself, I'm a pharmaceutical physician by training and thus spent most of my professional life in pharma, starting off in R&D in the late 80s. So today's focus of the webinar will be on metabolomics applications in pharma research and development, and only to a lesser extent on very detailed technical specifications. In the later Q&A session, of course, if there are very specific technical specifications, I'm happy to have Fan with me, who will be able to answer also very detailed technical questions on our technology platform. Let's go to the agenda. So I will give a quick introduction on the background of metabolomics, even though I anticipate that most of you are quite familiar with the subject. I will then explain the technology platforms we have at hand today, given the merger of Biocrates AG in Austria and Metanomics Health in Berlin. And then we'll quickly move into applications. We have 4 chapters. I will start off with a couple of in vitro examples published in the literature of application of the technology in discovery. We'll then move to the in vivo part, preclinical development. I have a couple of examples on applications of the more targeted assays and kits we have in the clinical development stage, and then also 3 examples on marketed pharmaceutical. Okay, so let's start with the background. Now, on site in Berlin, we are active since 1998 with a profound experience in gene function analysis, systems biology projects, combining genomic and metabolomic work. And we've realized that metabolomics is not only closest to the phenotype, but that our insights into biochemical principles, i.e. the changes in the metabolome, help us to understand the complexity of physiology and pathophysiology. So in other words, we therefore see metabolomics as the sort of Rosetta Stone of systems biology. Another important point I have learned since I'm with Metanomics Health in 2009 is that metabolomics is fully complementary and not competing with other omics technologies. And that's something which we should keep in mind. Moving on, I have two quotations from two Metabolomics pioneers because metabolomics is rapidly moving from exotic technologies for techies towards a reliable standard research and development tool. On the one hand side, I have a quote by David Wishart from the University of Alberta in Edmonton, Canada, from one of his Nature reviews. And then on the industry side, Donald Robertson, who works for BMS in Princeton in the United States and is there responsible for applied and investigative metabolomics in drug development research. And basically what they state in their reviews is that recent advantages, and that means advances over the past 10 years, the introduction of targeted assays, full quantification, the availability of kits, has led to the fact that metabolomics is becoming a standard technology in drug development, and that it's also coming closer to clinical chemistry performance. To a company with the merger between Metanomics Health and Biocrates completed January 3rd this year, we have now operations on two sites in Innsbruck in Austria, headquarters, and then the established Metanomics Health site in Berlin. And we have complementary heritages where I can say we are combining the best of two worlds. So one could say German engineering and perfectionism on the one hand side, and Austrian entrepreneurship and pragmatism. We have expertise on both sites since 2003, so more than 15 years. But with our teams, we have hundreds of years of experience with metabolomics. And we offer a range of options for our partners in industry and academia, from targeted assays via kits, or we introduced the first fully quantitative kit already in 2008, and untargeted, unbiased global profiling, both for service projects for our customers, but the kits also for captive or do-it-yourself use. Since we're all active in science, high-impact publications are the, so to say, Olymp of science in academia, but also highly valuable when it comes to you, our partners in pharmaceutical research and development. Here's a selection of more recent metabolomics publications from the years 2016 until early this year in 2018. Most of these publications are based on preclinical and clinical work with the P180 and P400HR kits. We've ranked them by impact factor for the gold, silver, and bronze medal. But overall, we and our partners in academia and industry have published more than 1,000 papers on metabolomics in peer-reviewed journals. So our technology has been widely used in order to bring research and development forward. Technology platforms, that is the next chapter. Today, with the successful merger of Biocrates and Metanomics Health, we can offer our partners 4 technology pillars. So we can offer by far more than a one-size-fits-all approach. With the merger, combining expertise and experience from both sides, we can now offer solutions truly tailored to your specific needs, which may range, as we see on the top of the slides, from early discovery and drug research to everyday patient care. Starting off on the left-hand side, global unbiased metabolite profiling for hypothesis discovery and hypothesis building, targeted and quantitative kits for captive use by our partners or also in service projects, targeted assays, for example, for steroids, catecholamines, eicosanoids, both the kits and the assays for hypothesis confirmation. And then more recently, based also on our experience on the kit side, but also when it comes to developing new diagnostic biomarkers, we offer co-development of diagnostic kits, companion diagnostics for pharmaceutical partners. What is important further to robust and reproducible results, we do offer our partners to transform metabolomics data into true information. So we have we give you support when it comes to project design, biostatistical analysis, and the plan for that analysis, and of course biomedical data interpretation on top of data-only projects. Let's move to the technology in examples a bit more specifically. I will show you the workflow of our global profiling, which is the backbone of our technology platform in Berlin, the previous Metanomics South site. Let's just go through it. We receive samples from our own internal programs or from our partners. Metabolites are extracted, proteins are precipitated, and then we separate into a lipid and a polar phase. Each of those phases is measured via GCMS and LCMS-MS. So in total, each sample is measured at least 4 times, polar and lipid phase via GCMS and LCMS-MS. Then, of course, a profound QC is done. We receive robust raw data, which we then link to our database, which contains more than 2,000 annotated metabolites, more than 2,000, as we call them, known unknowns. And as a result, depending on the matrix, we report between 400 and 700 annotated metabolites from more than 30 ontology classes. But of course, this depends on the matrix. I give you one example here. Human plasma or serum has less metabolites in it measurable than, for example, liver tissue. On a typical human plasma sample, we will report round about 450 metabolites. In plasma and serum, we can quantify 400 out of these 450 metabolites in order to make study-by-study comparisons or center-by-center comparisons possible. And what we, for human plasma, also offer, since we've realized that very often pre-analytical artifacts have a detrimental effect on metabolomic analysis, we offer, as part of a usual service project, a quality control of the samples, which gives our clients, sample by sample, a clear indication whether any pre-analytical confounders should be taken care of. So that's the workflow of the backbone in Berlin, global, untargeted, unbiased profiling. Moving on to the heritage here in Innsbruck, as I'm actually sitting in Innsbruck today. Biocrates has set new standards in targeted metabolomics with the first introduction of the first fully quantitative kit already in 2008, as I have said. We today offer for the broader profiling 2 fully quantitative kits, the P180, which is operated or done on a triple quad, and the P400HR, HR standing for high resolution, basically an expansion of the P180 kit for the high resolution instrumentation. And the major addition of metabolites is on the lipid side below. The kits, since they are fully quantitative, allow comparability of metabolomics results between studies and centers, and we're also constantly kicking the tires of our kits. So we have an ongoing program of worldwide ring trials with, on average, 6 to 12 participating core labs. And we've seen, and that has been published, a very low analytical variability comparable to standard tests. In addition, we have, and that's just a selection, a number of targeted kits. For example, the Biocrates bile acids kit, on which I'll show 2 examples later in the application section, the sterol kit. What I'd also like to highlight on the right lower quadrant of the slide is the MXP Energy platform, which is the second backbone of our technology platform in Berlin. Here we measure highly reactive metabolites with extremely short half-lives, such as phosphorylated nucleotides and cofactors, such as NADPH. We measure, depending on the matrix, again, 120 or more highly polar metabolites, so it's a fairly broad energy platform here. It's applicable to cells, in vitro research, for example, cancer cell line profiling or mode of action elucidation are typical applications, but it's also applied as a standard technology when it comes to improving biologics production in E. coli or other biological systems. Since it's very important for this platform, because of the short half-life of the metabolites, we offer our partners specific cell sampling guidelines, both for adherent and for suspension cell culture, and especially when it comes to biologicals production, we also offer our clients an equipment kit for separating cells and suspensions in seconds in order to make sure that in the end we do not measure artifacts but true biology, in order to improve time yield, space, and biologicals production. And then we have a number of additional very specific assays and kits available. Icosanoids, I've mentioned already, oxysterols, catecholamines. So basically something which will cover 99 to 100% of all your research challenges and questions. Now, this visual schematically depicts the breadth, width, and depth of our Metabolomics platform in Berlin. We basically cover all major metabolic pathways with our global profiling, and as stated earlier, we detect between 400 and 700 metabolites with our global profiling platform alone. The particular strength I would like to highlight here is the wide coverage, both of non-polar and highly polar metabolites. So that's a unique strength here. And we achieve that via the combined application of GC-MS and LC-MS-MS. You remember the first slide with the workflow. Each sample is measured both via GC-MS and LC-MS-MS, and that allows us to get a broader coverage than, for example, going via an LC-MS-MS approach alone. Now, characteristic core metabolic pathways and their fingerprint changes provide us information on physiological phenotype, on diseases. Already at very early stage, we see that in our clinical biomarker program that one strength of metabolomics as a technology is that since the metabolome is very sensitive, highly sensitive to changes, that we can capture these changes, whether it's disease or treatment effect already at a very early stage, and also classical and very typical toxicological fingerprints for target organ damage, for example, of new chemical entities. Now, which platform, which assay, which kit to use for which scientific questions? On the top of the slide, I've tried to show the classical value chain of pharmaceutical R&D from discovery over preclinical development, tox, clinical development approval. I've added in manufacturing because of the energy platform and its use as a standard technology to improve time, yield, space, and then market. And as a rule of thumb, if you look at the bottom of the slide, as a rule of thumb, you can say unbiased global profiling earlier in research when it comes to hypothesis building, when there are still off-target effects unknown of new drugs, we would rather recommend a broader approach via unbiased global profiling, whereas as we move on in the development phase, there is an increasing need for quantitative targeted profiling and the kits. So that's the rule of thumb, unbiased global profiling early on in development, in research for hypothesis generation, and later on targeted assays and kits to confirm and validate the hypothesis. But there are exceptions to the rule. I've also got a couple of applications, specific applications. I will not drill on each of those, but there are exceptions to the rule. I will show as the last application example a study which we did for a pharmaceutical client in a phase 4 program of a new chemical entity in respiratory disease, where the idea was to substantiate a positioning of the drug which is actually a me-too drug in this case, and the client wanted to show substantial off-target beneficial effects of the drug versus the established compounds in the class. But that will be described later. So there are always, as always, exceptions to the rule. Now we're moving to the main part, and that's the applications. And I'll start off with a couple of examples in discovery. So in vitro research and application of metabolomics. Okay, the slide moves on. So this is actually a good example showing the value of our broad energy platform in cancer cell line profiling or in vitro mode of action analysis of new cancer treatments. In this case, the question was by a Scandinavian pharma company, mode of action elucidation of a new chemical entity. Now, for confidentiality reasons, the NCE data cannot be shown, but I can show you the data of the generic API at which we looked in addition. As you can see on the upper left quadrant here, PLSDA loadings plot, and you see an upregulation, a striking upregulation of DUMP, and a downregulation of upstream and downstream metabolites of DUMP, which indicates that the mode of action of the comparator drug is the inhibition of thymidylate synthase. So you see in the graph, which I've now clicked on, the steep increase of deoxy-UMP concentrations by a complete blockade of thymidylate synthase and the downregulation of upstream and downstream metabolites. Now, the NCE in this experiment proved to work along a different mode of action, interference with the mevalonate pathway, despite very similar phenotypic effects on the viability of the cancer cells, in this case, DU145 prostate cancer cells. As I said, the other standard application of the energy platform, for which I have no time today to show examples because the focus is more on pharmaceutical development and precision medicine, is the application in biologicals production in an effort to increase time use space. Now, the next example is an example of a tailored metabolomics method development for a pharma client. So the question here was, by our client, can you expand the energy platform to include all gemcitabine metabolites? So the prodrug, the active, and degradation metabolites, total of 8. And the background for this client was, well, how are these metabolized in vitro systems and, of course, in tissue and in whole organs, whole organisms? What may be the origin of acquired gemcitabine resistance, and can we develop biomarkers suitable for patient stratifications? So this is what we did. We added the 8 metabolites and the prodrug itself, or the 7 metabolites and the method. This is actually published in a white paper already from 2012, and if you're further interested into that as one of the examples of a tailored specific method for a client, which we've now opened, of course, in agreement with the client towards everybody. You can download the white paper from the Metanomics homepage. Moving to the next example, this is a publication now from Caiola and colleagues from the Mario Negri Institute in Italy, published in Oncotarget 2016. The challenge here was to understand the background for drug resistance in non-small cell lung cancer. Here, tested via PI3K inhibition. 2 compounds were given, as indicated on the left hand of the slides, and it's an in vitro experiment. And we looked, or the authors here looked at KRAS mutations and wild type. Since KRAS mutations are considered negative predictive factors for non-small cell lung cancer chemotherapy. Now, in vitro, 2 compounds were given. On top, you see the cell viability with the first compound. On the bottom, cell viability. Second compound, And as you can see here on the viability, and likewise for cell growth and cell cycle distribution, there was actually no difference between KRAS wild type and the KRAS mutant. Now, if we look at the metabolic profiles in this in vitro experiment, which I can show you on the next slide, on top, the KRAS wild type, on the bottom, the graphs for the KRAS mutant. You can see that PI3 kinase inhibition in the wild type suppresses the serine metabolism. Now, serine, as we know, is required for synthesis of nucleotides, proteins, and lipids, so it's a very important essential amino acid. So that seems to be the main mode of action in the wild type non-small cell lung cancer cells. Whilst if you look at the bottom, PI3K inhibition has an impact on the aspartic acid, glutamine, and glutamate metabolism, which are all three impaired, indicating in this case with the mutant a totally different molecular mode of action via glutaminolysis and the TCA cycle. So we can clearly differentiate via our technology or the application of our technology. In this case, the P180 kit was used on a triple quad. We can clearly differentiate the molecular mechanism of action of chemical entities in different systems. Moving on, we are coming to the preclinical section, more in vivo research, still in the early development. This is a study which was published in 2015 from Stanley and colleagues, basically from David Wizard's team in Edmonton, the University of Alberta. And it addresses one of the early development challenges. Well, can we somehow explain the mode of action of new chemical entities in preclinical models, for example, via knockout mice, classical model? Now, this is a study which was done in knockout mice, COMT knockout mice, so catechol O-methyltransferase knockout mice, which in these mice leads to hypertension, proteinuria, and fetal growth restriction late gestation. So basically, it's a model which is mimicking preeclampsia. Now, sildenafil is known for its effect to increase fetal growth in preeclampsia models, but it's not really understood so far how this works. And so what you see is that in the COMT model plasma, acylcarnitines, a number of acylcarnitines were increased, glycerophospholipids decreased versus control, sphingolipids decreased, and kynurenine also increased. Now, by application of sildenafil, most of the changes in this knockout mouse model were reversed. So we see a reversal of the metabolic profile under sildenafil. And we see that in conjunction and parallel to the positive effect of the compound on fetal growth. So in an association between the metabolic impact of the compound and in this case, physiological response. Another application, and this is now where the authors used the Biocrates bile acids kit. This is actually a publication from Slopjanka and colleagues from Biopharma, published in Toxicology 2017, addressing drug-induced liver injury. Now, drug-induced liver injury, as you all will know, is the most frequent reason to terminate a new chemical entity development in late clinical phases or early market withdrawals. So it's a true challenge in late development, and it's often recognized too late. Now, in this study, our partners applied the bile acids kit in vivo in rats. And the model they used is a methapyrilin-induced, drug-induced liver injury. Now, as you can see from the PLSDA plot, the separation between the rats treated with grams per kilogram versus the placebo-treated animals, there's a strikingly clear separation of the two groups. And this is actually driven by a broad range of primary and secondary bile acids, which are increased in plasma in the treated animals. And one of the core findings here was that, of course, drug-induced liver injury was also monitored in this study histopathologically and via the classical liver enzyme tests. The striking point is, and we've seen that in other toxicological studies addressing DILI as well via metabolomics, that the changes seen, so the increases in primary and secondary bile acids were persistent also in the post-treatment washout phase, and thus more consistent with the persisting histopathological changes than the classical lab chemistry tests such as the liver enzymes. So a valuable addition to the armamentarium in toxicology to look at drug-induced liver injury. Another example on a topic which is one of the hot topics in metabolomics, not only in metabolomics, is that the metabolomic analysis of the intestinal microbiome is an increasing applications in the area. This is a publication by a Canadian group from Toronto, from Musaki and colleagues, published in 2016. Now, they looked, again, in this case, in stool. So it's the analysis of stool looking at, let's say, changes driven by differences in the gut microbiome. And the model they looked at, or the clinical case they looked at, is that changes in the intestinal microbiome may promote NAFLD, so non-alcoholic liver disease. And we know that dysbiosis in NAFLD is shown by altered counts of Bacteroides and Proteobacteria. Now, what the authors here did was comparing biopsy-controlled NAFLD in NASH patients with matched controls. And they looked at the bile acids in stool, again, primary and secondary bile acids. There was a very significant increase in overall bile acids in stool in NASH patients. This is actually not shown in the graph here, but what you see here on the graph is a decrease of the, so to say, evil secondary bile acids. For example, the oxycholic acid and litocholic acid in stool in NASH patients versus healthy, and a decreased ratio of primary over secondary bile acids in stool. So more secondary bile acids, which have a negative connotation, of course, as you all know, in the enterohepatic circulation and the bile acid pool in NASH. Now, this is one example of applying metabolomics, a targeted kit towards stool analysis, thereby indirectly looking at the impact of the gut microbiome. But what I would like to highlight here is that we have also fully validated, on the one hand side, the unbiased global profiling platform and the volatiles platform we have in our hands for short-chain fatty acids and volatile amines. We have validated both of these in addition to the bile acid kit for stool analysis, thereby allowing a very broad and deep analysis of changes in the gut microbiome in a number of diseases. So far with the preclinical development, let's move to a couple of examples from clinical projects. Now, as we all know and appreciate, the metabolome is highly conserved. So core metabolic pathways are very similar even between plants and mammals, and this is not the case with metabolomics technologies, as you all know. So metabolomics findings are highly translational from animal to man, and therefore particularly useful when it comes to pharmaceutical development, which usually starts in vitro, then moves on to animal experiments, and then eventually in a successful case ends up in human studies. Now here's a publication from Ang and colleagues from Genentech from 2016. with a couple of academic collaborators from the UK and Australia. And what they actually did was an experiment in mice xenografts of prostate cancer and glioblastoma, and compared that to the phase 1 clinical results. So what you can see, and I've blocked part of the slides, which we'll be building up in a minute. What you can see that in plasma concentrations of 26 metabolites, including amino acids on top, acylcarnitins and phosphatidylcholines were decreased. So the blue color here indicates a decrease. That's an original picture or graph from the publication in mice bearing P10 deficient tumors, so phosphatase and tensing homolog deficient tumors, compared with non-tumor bearing controls. So a whole range of metabolites decreased. Now, with a class 1 PI3K inhibitor, pictilizib in this case, in mice, shows over time, so this is after 1, 4, 6, 8, and 24 days, a reversal of the pattern we see in the native case. Now, they also looked, as I said, into phase 1. So here are in parallel the translationability of metabolomic results from animal to man, a phase 1 dose escalation trial with 3 dosage ranges here, and you see dose-dependent over time a reversal of the pathological pattern we otherwise see in parallel to the animal experiment. The other interesting finding, and that's highlighted via these 3 green branched chain amino acids, which basically confirms what is known for phosphoinositidine 3 kinase inhibitors, that they, as one unwanted side effect, lead to a pre-diabetic or diabetic stage in patients. So a nice example showing how translational the technology can be in pharmaceutical research. Now, the next example is published by a group from Amgen, Elizabeth Finger, not fully published as a paper, but published in 2017 in poster form at the American Society of Hematology. The manuscript is in preparation. Now, topic here was to look at acquired drug resistance in standard of care chemotherapy on the one hand side in acute myeloid leukemia, and also in diffuse large cell B lymphoma. So leukemia and lymphoma. We know that tryptophan catabolism, including the dominating kynurenine pathway, is involved in immune regulation, and that's why the authors here looked with the P180 kit with a particular focus on tryptophan, serotonin, and kynurenine from the same area, pathway area here. Now, the IDO-driven tryptophan catabolization of leads to kynurenine, and that has 2 effects on immune modulation. On the one hand side, T effector cells are suppressed, whereas stabilizing regulatory T cells, formerly known as suppressor cells, are stabilized. And what we can see on top, you see the results from healthy controls, so normals, versus newly diagnosed leukemia and resistant leukemia. You see an increase in kynurenine over the graph. On the lower chart, you see again normal controls or matched controls, and newly diagnosed lymphoma and recurrent lymphoma, and you see the same effect, an increase in kynurenine. Now, clicking on the slide once more, in partial remission and complete remission, we see a trend towards a decrease again to normal levels of kynurenine. And overall, we see a IDO activity which is decreased, and the ratio of kynurenine and tryptophan is decreased in remission patients. Okay, so that's another good example from the clinic. Let's move on to the last and final chapters. 3 more examples I would like to share with you. First of all, a publication by Bhattacharya and colleagues from the University of Arkansas. One of the authors is Rick Beeger from the FDA, multi-center study looking at kids, normal controls, kids who received regular doses of APAP, paracetamol, and APAP intoxication with an administration of more than 150 milligrams of APAP per kilogram body weight. Now, APAP toxicity is the most common reason for poison-related hospital admissions in the United States and for acute hepatic failure in the world. So it's a challenge, a real healthcare challenge. Previous studies in cell cultures have shown that APAP has an influence on phospholipids as building blocks of cell membranes and also in their function as cell signaling molecules. But so far there were no patient data on this available. So what they did was a study in children looking at controls, therapeutic and toxic doses of APAP, and as you can see from the PLSDA plot, you see a very clear separation of the 3 groups. Now the PLSDA separation has a number of drivers. Basically, it's driven by 25 metabolites, 22 of which are phosphatidylcholine and lyso-PCs. Now, looking at control versus therapeutic, you see already differences. And if we look at the overdose, the toxic dose of APAP, we see that APAP in the higher dose kicks off structure-dependent changes in phosphatidylcholines. The short chain PCs go up in their concentrations versus the long chain and very long chain ones which go down. And that helps now to better understand the mechanistics behind APAP-induced liver toxicity. Another clinical example, a routine question for all of you active in drug development today is prediction of response. And this is a study, an exploratory study by Miolo and colleagues, published in Oncotarget 2016. The group is from the National Cancer Institute in Aviano in Italy. And the question was here to predict response of a combination therapy of trastuzumab and paclitaxel in HER2-positive breast cancer patients. Now we know that even though we have the HER2 test available, the proportion of patients responding to therapy is still in the 30 to 35% range only. Now, what they did is they applied the P180 kit on a triple quad, and as you can see in the graph in the right upper quadrant, good responders, so GR, and poor responders, PR, a very clear group separation. Now, what in this case does drive the group separation? The pretreatment measurements with P180 revealed a reduction in the essential amino acid tryptophan and an increase in spermidine as positive predictors of therapy. And as you can see from the AUC curves here, the top curve, the blue curve, shows the ratio of tryptophan over spermidine, which gives an AUC of 93%, so a high level of prediction for the response to therapy in these patients. So this could offer, in addition to the HER2 test, an easy test to stratify patients towards responders and non-responders prior to the initiation of therapy. Last but not least, the last example from the clinic, and that's the example which I stated earlier as the exception of the rule. These are data hot off the press, so therefore the graph is not optimal. I have to commit. So it's the application of our global unbiased profiling platform in a phase 4 trial looking for beneficial off-target effects of a new compound. These data are actually this week, as we speak, presented at the meeting of the American Thoracic Society in San Diego. by Klaus Rabe from a pulmonary lead clinic in northern Germany on behalf of the Robert trialists. The study was originally planned with Nycomed, which is now part of Takeda, and was carried out and funded also in cooperation with AstraZeneca. Now, the objective, as I said, was here to substantiate scientific evidence of a positive cardiovascular profile of a Me2 PDE4 inhibitor in obstructive lung disease in the clinic. In this case, roflumilast is the compound, which is added to steroids or beta agonists and COPD. Now, shown here are changes, and these are increases and decreases. So the number of changes of metabolites by metabolite class after 6 and 14 weeks versus baseline. And what we see with the bars above complex lipids, you see a sustained reduction in complex lipids over time, which parallels the changes seen in HDL and LDL cholesterol and total glycerides here. And what you see on the amino acid side, the authors have seen a very clear increase of arginine, which is increased and which plays a role in the vascular nitric oxide production. So this study in 158 patients helps to substantiate evidence allowing a positioning of this Me2 compound in its class via a positive additional cardiovascular profile on top of the primary action in COPD. Now we are coming to the summary here. I hope I've been clear enough to convince you that we have now a toolbox in our hands which allows us and our partners to very specifically and not in a one-size-fits-all approach to address questions along the pharmaceutical R&D value chain. We offer these tools both as data-only packages, but also given the full statistical analysis in order to help you to transform data to true information. And with that, I'm coming really towards the end with this graph here, because very often the question is, well, what is better, broad untargeted profiling or targeted profiling? We strongly believe that each has its place, and we at Biocrates and Metanomics Health have the possibility to offer you both, and can you give a true and honest counseling towards using the right technology for the right development challenge. And with that, I'd like to finish off the presentation. Here you find my direct email address, at least, not my phone number, and I'm happy if you, after this presentation, first of all, we'll have a Q&A session, of course, but if you, after this presentation, if there's any interest, you get in touch with me, and we can start a dialogue via email. And with that, I also would like to thank all team members here in Innsbruck, where I'm today, and in Berlin, who've made this webinar and presentation possible, and would now suggest, Martha, we move on to the Q&A session.
Yeah, of course. So thank you very much for that insightful presentation, Tim. I hope our audience found it of great interest and value. If you do have any questions, you continue to submit them using the Q&A tool on the right-hand side of your screen. So we've received a number so far, so we'll get started with those. Tim, our first question asks, are there any clinical or diagnostic products already available on the market?
Yeah, what I haven't mentioned today is we have both from the Biocrates heritage, but also from Metanomics Health, a broad biomarker program, as we call it, where we try to establish and develop new diagnostic markers in areas of high medical need. But none of those has really hit the market. We are in discussions with clear laboratories in the United States, and there's also raising interest in China to bring these drugs to market before they actually become IVD diagnostics. But to the question directly, No, we have no diagnostic markers put to market yet. That's still to come.
Great, thank you. Next, we have someone asking, are you experienced with clinical trials?
That's a very good question. I think I would like to answer that from two ends. On the one hand side, as stated, we have our own proprietary clinical biomarker program. So we do run clinical studies with our academic collaborators when it comes, for example, further elucidating our diagnostic marker for the early detection of pancreatic cancer. We work with CROs, so we have own hands-on experience, but we on a regular basis counsel our clinical partners in academia, but also in the industry on their clinical trials when it comes to recommending the appropriate assay kit or profiling. What is also important, the quality marker, which I have briefly mentioned when we went through the workflow of the global profiling, which allows us to really assess the pre-analytical quality of samples from clinical trials, is also an important tool. So we have a number of touch points with clinical trials, and we do run clinical trials ourselves. So I think we have a good understanding where the major challenges are and usually can engage into a fruitful dialogue with our clients on the challenges they might experience along a phase 1, phase 2, phase 3, or even phase 4 program.
That's great, thank you. How many metabolites are typically reported for a human plasma sample?
That's also a good question. It really depends on the method applied. As I've tried to show, is we have a very broad toolbox available. If I take the global metabolite profiling, which is the backbone in Berlin and the starting point of all of our activities at the Metanomics Health site in Berlin, we have really jump-started the method here, where we now report around about 450 metabolites in plasma. The highest density of metabolites you might find in a tissue such as liver tissue, where it goes up to 700 metabolites which are reported. And that's the global profiling. And then we have targeted assays. Let me just take the bile acids kit from Biocrates here in Innsbruck. There we report up to 20 bile acids. So it really depends on the method chosen and the matrix. Now the question here was how many are reported in plasma. Broad profiling, 450, 400 out of those fully quantified. The targeted assays and kits are slightly less than that, or depending on down to 20, for example, with the bile acids. If that's an answer.
Great, thank you. So we have a few questions together here. What are the options to run the analyses? Are all the assays offered as service only? And are there any core facilities where the analyses could be performed?
Now, the global profiling and the assays have to be run in-house. So they are performed either in Innsbruck or in Berlin. And we provide these as a service. Now, the service can range from a data-only package, where we provide our partners with a basic statistical analysis, down to a fully fledged project where in the end our partners get a full report, a state-of-the-art univariate, multivariate biostatistical analysis, and a biomedical data interpretation based on indication experts. Now, I cannot read it anymore. What is the
Oh, yeah. Hi, Tim.
Okay.
Can you just repeat
so
sorry.
The global profiling are run as a service only, different packages, as I said. Now, with the kits, these are easy to be used. Once installed and trained by our technical staff, you can run them on site. If you have a triple quad available, the P180 and a couple of the very targeted assays are possible to be done in a captive manner or do it yourself. We also have core facilities. For example, these kits can be run by the Helmholtz Institute in Munich and by the group of Dave Wishart in Canada, as an example. And we can run them in-house. So depending on timing needs, specific requirements, we have a true choice also here, which we can offer to our partners.
That's great, thank you. And following on from that, on which instruments can the kits be run and what other requirements are necessary?
Okay. So kits basically, let's focus on four of them. The P480HR kit, as the name indicates, HR for high resolution, not human resources in this case, can be run today on the ThermoQ Executive instruments. So for the time being, that is the technical platform where the kit is validated. The P180 kit, so the P400 was launched late last year. The P180 kit, which measures around about 180 metabolites, fully quantifiable, is established on a broader technology platform. So it runs, for example, on Waters, SCIEX, and Thermo triple quads. The steroid 17 kit, where we measure up to 17 steroids, can be run on Waters and on SCIEX. Very important to note, for the time being, the steroid kit is not available in the United States. We are working on that, but for the time being, it is not available for IP reasons in the United States. And then last but not least, the bile acids kit, which I've explained with two examples, also including the gut microbiome analysis here, also established on Waters, SCIEX, and Thermo instrumentation triple quad.
Great, thank you. We've got a question here asking, what is the typical duration of a service project?
That's a good question. So let me just try to, let's say, describe a typical service project, because the duration, of course, depends on a number of things. How many samples have to be measured? There's a big difference if we get a project kicked off with 100 samples versus projects with 2,000 or 3,000 samples. Of course, analysis there takes longer. It also depends on which assay or kit has to be used. A typical project, let's say 100 to 200 plasma samples from a clinical trial. We guarantee our clients that the analysis starts at least within 4 weeks after receipt of the samples with the complete documentation. With the global profiling as an example, the analysis is finished within 1 to 2 months. The interim report is then produced, and the final report is available 3 to 4 months after receipt of the samples. Now, this is the standard. Of course, sometimes there are important time windows for our clients, so it always makes sense if there is a need for a faster performance of a study to take that up with our business development managers in the field, and we'll always try to accommodate specific urgencies of our clients. But let's say the typical project until the final report is finally signed off, also and accepted by our customers, 4 months. Now, data only, for example, cuts out around about 2 months at least. So that's maybe in between 6 weeks to 8 weeks for a data only project. So there's a variation. Yeah.
That's great, thank you. So why should the MXP Energy platform only be applied to cells, tissue, and whole blood? What about blood plasma?
Well, I mentioned that only briefly initially. It's a specific platform where we measure metabolites, highly polar metabolites with very short half-lives. phosphorylized nucleotides, NADPH. Now these have in a biological matrix a half-life of a couple of seconds. And in order to, in the end, measure these metabolites in biological and meaningful, in a meaningful setting, these samples have to be immediately snap frozen. We have guidelines in place, which we provide to our partners in order to make sure that they are in a position to really prepare these samples within a few seconds, even for cell cultures, for example, in suspension, that is necessary. Because otherwise, you can always measure these metabolites, but if the sample is only put into the freezer 5 minutes, 10 minutes after it has been taken, you can measure artifacts and you will no longer measure biology. Now, plasma is a matrix which takes its time to get. So to prepare plasma from blood takes too long in order to really in the end have a sample available where it makes biological sense to actually measure these energy metabolites in that matrix. So the limitation here is really how fast can I prepare a sample, and that limits the meaningful application of this high value platform. So that's the reason behind it.
Fantastic.
I think we can take one last question. I think we.
Yeah, I was going to say, I think this is the final question, I think. When should I choose the MXP Global Profiling when there's the P400 HR kit instead?
That's a very good question. I mean, it depends on a couple of parameters. For example, unless you have a high-resolution thermal instrument in your lab, you may not use the P400 HR kit. So it depends on your technical capabilities in the lab, whether you can actually do the kit yourself. We would offer, however, the kit also as a service, so we could do that as a service project versus global profiling. I think it really depends on the project specifics. One of the differences between the two is there are many differences, and we would counsel our partners really in detail on the pros and cons of each platform. One of the big differences is if there's a particular interest into polar metabolites, the coverage of the global profiling is broader. And in plasma, we have a tendency, while we see 450 metabolites, for example, with global profiling versus the P400, so there's slightly more metabolites in that particular matrix. So again, as in many of the other questions asked beforehand, it depends on the project. I think the good news for our partners out there in academia and industry is that given the merger of Biocrates and Metanomics Health, they now really have from the same supplier, because we're one company now, a true choice between all technical tools available.
That's great, thank you. I'm afraid that's all we have time for today, so we'll address any other questions offline. But if you do have any questions for Tim, please feel free to send them in to webinars@bioanalysis-zone.com. So I'd once again like to thank our wonderful presenter, Tim, as well as you, our listeners, for your time and your questions. So if you'd like to let us know your thoughts on today's webinars, please feel free to complete our feedback survey in the resource widget or to tweet us at BioAnalysisZone. Don't forget to visit us at www.bioanalysiszone.com for more webinars. Thank you again for attending, and goodbye.
Also from my side, thank you very much for attending today, and hopefully you will get back in touch with us in the future. Bye-bye.